You are saying.
So, a very beautiful morning to all. So, first of all, on behalf of Director NIAB and from the NIAB fraternity, we welcome our today's honorable guest, Dr. Priya Abraham, Director NIV Pune, and Professor Sharmila Bapat from NCCS Pune. Madam, a very warm welcome from NIAB and to the city of Pearl, Hyderabad. So this is a very memorable day for all NIABians. There is a lot of unforgettable and cherishable memories that are attached to this day and those earlier days for all of the NIABians who have contributed immensely in the building of this institution and continuously contributing to the scientific journey of this institution. And every year we organize this meeting to remember those and also remember those contribution of the people and also enrich NIAB in its scientific path by learning from all the learned scientists and luminaries in India, calling them to our campus and sharing and learning from their knowledge and experiences. So today again, we are on that path. 5 years back, we moved to this campus from our temporary campus in Miapur. And here with us, none other than Dr. Subir Majumdar here to say a lot of story about those memories, how all the people, right from administration to staff, support staff, scientific members, and the student community contributed immensely in building this institution. And now I will call our director, madam, to introduce the story of NIAB science to our today's guest. Madam.
Thank you very much, Dr. Bapaditya. Very warm good morning to one and all. Well, both the speakers of this important day, Dr. Priya Abraham, Director, National Institute of Virology, Pune, and incidentally both from Pune, and Dr. Sharmila Bapat from NCCS. The scientist who is known for her outstanding stem cell work on ovarian cancer, and she will be hearing. My own colleagues, scientific fraternity, people from admin, my dear students, fellows, everybody in the hall. As Dr. Bapaditya has said about this story, we decided and chose let Professor Subir Majumdar elaborate about this day, and let me say a few things about the institute. This institute is 11 years old and started in 2010, and it started from University of Hyderabad. And it was surely, as I was talking to you both, Professor M.K. Barnes' brainchild. So this is one of the new generation Animal Biotechnology Institute. A very small place, the university is big, but we started with a very small place, and the journey which was traveled by the dedicated group of scientists, administration, and the students who could bring NIAB to this place where we are today. A temporary campus was set up at Miapur, And I'm sure people who came for stem cell workshop organized at that time remember that campus. There are a good number of students besides scientists and our administrative colleagues who have been holding hands in the journey, if not from University of Hyderabad, from Mayapur to the Gachibowli, this very campus which was inaugurated on 11th August today in 2018. Well, the institute is not that old, but in a decade's journey, institute could make enough dents into science, and this could become possible due to a very sincere, dedicated efforts of the scientists, the students, fellows, and administrative colleagues who have not left any stone unturned to support this group so science grow and move ahead. All this was not enough. DPT and the officials from DPT, a couple of them, if I could name, Dr. Rawat, Dr. George on that time, have really held hands to support the institute's science and the way forward. I was showing both our guests the chip, which was being generated. The story I have already shared with them. It is a pride of the institute. Besides that, we work in mandated areas which cover different infectious biology, different pathogens, and infectious biology group deals with it. We do have a reproductive biotechnology team, and we have a very specific group which is focusing on transgenic animals. And we are moving ahead as a way forward because it was a core area of the former director who has been working on this area, and we're looking forward to move with that. And along with, as the team moved on, people started joining, and they came with different fragrance, scientific fragrance. People came with the nano diagnostic model, nano delivery, genomic group, bioinformatics group became more stronger, and the reproductive biology people join, and different people join who started working on different zoonotic diseases. And DPT understood it, and they have given very focused flagship projects to the institute, under which only the chip came. That was a livestock genomics project, one of its own kind in the country. And another zoonotic diseases, as I said, the Leptospira group and the Japanese encephalitis virus to that flagship program. And a little more than a year back, this institute has started One Health program, which is a very prestigious program of the institute, coordinated by Dr. Subir and led by Dr. Hegde. Therein, we have a very fine amalgamation of medicals, veterinary scientists, and a basic biologist. And the group did not leave a very important partner, the wildlife group, because without that, Dr. Priya will really agree that if we leave them, the story will not be complete. And we were discussing last evening about it, several more steps to put on. The teams which are very focused, and now it takes time in veterinary animal sciences for the results to come. In most of the biological sciences, now the fruits are kind of coming. Different diagnostics which have seen the light of the day and being commercialized, or in a verge of commercialization. Couple of patents could be submitted, and maybe in another year or so we will have them. Then we are working with the specific molecules which have been identified towards different vaccines. We are planning, if not 4, at least 3 are in our on our cards. And likewise, we also we are also looking forward to strengthen the area of nutrition and its collaboration with the reproduction so that the production can be improved. This is in a nutshell about the research, ongoing research of the institute. It's a very happy institute, having very I mean, scientists, they are masters in their own area, And researchers and fellows, they are the ones who are the actual life of this institute. Besides that, we have a very dedicated administrative team, always in a hand-holding way we move. And about this day, before I invite and Dr. Subir says, it was 11th August when everybody moved to this new campus. It was not a very easy task. It was a Herculean task. The team led by Dr. Subir and all the scientists, and I would fail if I do not take Mr. Harjit's name and his team. Besides, all the students who have traveled from Mayapur to this place have really joined their hands with the scientific and administrative team, so that we are here to celebrate our Institute Day. I personally, on my own behalf and on behalf of all of us, join Dr. Bapaditya once again to welcome you both. Thank you very much for making it, and we look forward. You request or I request? So we invite Dr. Subir to please come and highlight about the Institute.
Good morning. Welcome to esteemed guests. I know Dr. Priya, as she is very famous in India now and has a lot of awards. And she is a great virologist, no doubt. And Sharmila is a very old friend. Sharmila is not old, but we are friends from a very long time. And she's a forerunner of the woman scientist. She has really, and keep on fighting for several causes, very straightforward. And that's why I have very good relationship with her. And first and foremost, I congratulate everybody sitting in audience and not in the audience, our past students who have gone from here, and all project staff who have left NIAB, either to get a better opportunity in life or to get a job or get married. Everybody's contribution I recognize and I congratulate them on this day, and I thank all of them. Because of this, we can see this institute grown up into such a nice, not physically nice, but morally nice, culturally nice institution. So if you get a chance to go anywhere, either in hostel or on roads, you will know that we spend a lot of time with students, because I thought this institute is being made Animal Science Institute. No student was ready to come here, because before 7 years, if you would have called इधर आके lecture दीजिए, आपको लगता है क्या गाय भैंस के institute में जाके क्या lecture देंगे? I mean, it's just all students want to work on cancer, biology, tuberculosis, and virology, vaccine development, and all these things. So we had a very tough time. So I respect all the students who joined NIAB in the first, second, third batch very much. I do not know how good or how bad they are, but I think without them, this institute wouldn't have stood erect. As Madam has already said, this is an Institute Day. It is very important to remember people because of whom this institute is erected here. It was Dr. John George, and definitely our great leader of this country, who is no more, Professor M.K. Bhan, who decided to make an animal institution. So whenever a new institution is made, it goes to the cabinet, and from there goes to all ministries. All ministries have to say, such kind of institute doesn't exist in our ministry, so you can make it. Obviously, there was no choice with ICR. They cannot say we do not have such kind of institute. They have 32, 34 institutions related to livestock research, livestock science, extension programs in the farmers. So they declined and denied. And Dr. Bhan, I still remember Dr. Rawat tells the story that he took his coat and threw like this on the ground and said, "Who are you? What is your name?" I will not name that gentleman. But then later on, those people came to me and said, "We rejected your institute and we repent for it. We have come here and we have seen what you have made. And with the grace of God, the scientists who have joined here were so bubbling, and we are so frank you cannot imagine the way we have developed and groomed ourselves, that there was no choice other than productivity to come. And we all understood, including students, that in animal science, we can do wonders if we do not prove that. Nobody is going to give us respect. Most importantly, scientists understood. And I always narrated to some of them this story, that two people who were working with chappal ka business, the sleeper business. So students have to learn from this. So their bosses, let us say Relaxo and Corona. Corona, what is the name company? Corona, something. There was a company, Corona or something, Jutega. So those two salesmen were sent to Arabian country with 10 boxes of slippers and sandals and shoes. You go and start the shop, open there. So after 1 month, one of them wrote back, this is a all Registan sand business. Nobody wears your chappal. So I have dispatched the 10 boxes back to India, and I am also coming in tomorrow's flight. So there is no scope. The other salesman wrote that this is all land of sands. Nobody wears chappal. Send me another 100 box. I'll teach them how to wear chappals here, and we will have a great sale. That's what we exploited from these institutions, that this is an open field. What ICR people are working on are not necessarily extraordinary modern biotechnology, which Department of Biotechnology wants us to do, and they are ready to support. We exploited that single thing, and with the grace of God, there are so many good things which we have done, which everyone praises in the country, and our existence has come into kind of getting advertised to every one of us. So I thank everyone who has participated, but it began with Professor Bhan and Dr. John George. An actual person who ran around was Dr. Arun K. Rawat, who is still with us in One Health as a consultant. His great experience, and they really loved the institute. Now, for example, this room was not a room. When I came here, construction was half the way. There were 2 lecture halls. So the walls were erected up to this stage. I broke the walls. And they said, sir, ये क्या कर रहे हैं? Design में नहीं. I said, नहीं, so तो इसको auditorium बनाओ. कह रहे हैं, सर, ये दोनों lecture hall है, design में government से मतलब कुछ नहीं. Life में ये भी lecture ही हो रहा है. So it will still be lecture hall. Out of other than 2, it is a 1 lecture hall. Life में कभी दोबारा chance नहीं मिलेगा auditorium बनाने का. So height was very less. So I said, बस इतना height रखो कि stage पे बच्चे भांगड़ा नाचे, तो उछलने के बाद सर पे नहीं लगना चाहिए, बाकी चलेगा. So, like that, jokingly and all, I got all people involved, and we built this institute with great passion. With the grace of God, Jagdish, Santosh, Harjeet, Pawan, and so many of other colleagues I cannot name, and their whole group, they were so wonderful and so nice. They got so activated. Ramesh is here somewhere? No. Electrical Ramesh is not there. So he's busy, I think. So, I activated all of them, and we got rejuvenated, and we worked very hard. All scientists who were there. There's a huge board like this, and everybody has some work. कोई gutter का इंचार्ज है, कोई manhole का, कोई दरवाजे का, कोई wall का, कोई carpet का, कोई पेड़ों का. So we did wonderful job, and we did half science, half work for building this institute, and this institute was built very nicely. With the grace of God, although we built the institute and we were happy, something very bad came in. I'll take 1 more minute to tell that. Is that Pollution Control Board cancels the license of this institute. Same thing happened to CDMD with Dr. Hasnain. And they fought up to Supreme Court. They lost the case. And they quoted on the file that like CDMD, this institute is within 10 kilometers of the drinking water body. It will have the solutions which are chemical and all in it. So this cannot be granted. I joined on Monday. Friday, naturally, I went back to my home in Delhi. And next Monday I came. Harjeet said, sir, worst thing has happened. My hands are shaking. I didn't call you. We have lost. This all half building is made and this is cancelled. I said, okay, ठीक है, तुम ले आओ. तुमने कुछ किया homework है, सर मेरे से जितना हो सकता मैंने किया, उन्हें ले आओ. Then within 1.5 to 2 hours we wrote a letter. Allah, चलो, तो कह कहाँ जाना है? Pollution Control Board. अरे सर आप क्यों जाएंगे? मैं जाता हूँ. तुम गाड़ी निकालो, चलते हैं. And you will not imagine, Madhuri is here and Satta is not here. So within, I think, 25-30 days, I don't know what I have done. They don't know what they have done. Harjeet doesn't know what he has done, but we played all tricks, including writing a poetry for the senior most IAS officer because he was a poet, making friends with him and and he's saying that yes, Sunil Kumar used to work under me, who was chairman of the Pollution Control Board, IAS officer. Then sitting with Sunil Kumar, he's saying rejected is rejected. And then I told him, madam, sir, हमको तो कुछ भी नहीं मालूम, हमें तो पैसे का ज्यादा लोभ था, ये पोजीशन हायर सैलरी की थी, तो मैं तो नौकरी छोड़ के आ गया. आप लोग IAS ऑफिसर है, you guys know how to run. We don't know how to run, and we are no, you are such a come sit on the sofa set. It worked. सोफा सेट में बिठाया और सर हम तो कुछ नहीं, हम तो रात के 10, 11, 12 बजे तक काम कर रहे हैं. इतना काम करोगे तो आपका तबीयत खराब होगा. सर मुझे देखिए थायराइड है, सेंट्रल ओबेसिटी है, बहुत परेशान है. माय वाइफ हैज थायराइड. देन आई वेंट ऑन हिज वाइफ, what she must be saying and how should he treat her and how I can help him. And he helped us to revert the whole phenomenon, and rest we know how we did. यहाँ तक कि मैं और हरजीत एक ऑफिस में घुस के क्लर्क से बात करके सैंक्शन हो जाने के बाद भी उस लेटर में मुझे एक वर्ड अच्छा नहीं लगा जो हमने अंडरटेकिंग लिया. देन माई सेल्फ एंड हरजीत वेंट, मैं से हरजीत मैं इसको रिप्लेस करके आता हूँ. तो हरजीत से सर आपके लेवल पे अच्छा नहीं दिखता है, आप बैठिए. फिर मैंने हरजीत को भेजा और वो कागज भी रिप्लेस किया हमने सैंक्शन होने के बाद, क्योंकि वन वर्ड वी हैड अ डाउट कि आगे चल के कुछ हो सकता है. So we have done wonderful thing, and this institute is here, and I am very, very happy that you have come and visited our institute. I hope you will go back and tell your experience to others, and people are very nice, and I am proud of each and everyone who joined after we moved into this institute, whether it is Ravindranath or Prem Kumar and his team and so many other people. Keshav, I can see some of them here, Shravan, everyone is so nice. I cannot name all of them, all students. And the only culture which I maintain, I personally feel that these are all the trees implanted by us, definitely by me. I love them, I nurture them. I can never be angry with them. I can never be unhappy with them. Whatever they do, my only expectation from them is, even if I survive up to 90 years, I must see in a newspaper that Dr. X has done this or Mr. X has done that. And I should feel proud that these are the people I nurtured well. So I have all good wishes to all of you. Grow well. And I sincerely pray to God that not only all of us, but Institute should grow well. I am thankful to Dr. Taru Sharma for giving me a chance to speak here today. And it is in her, she always says the baton is given to her. And I am very sure that she is quite capable and very hardworking. And she has a challenge to kind of a tough time to compete with me. And I give her all my good wishes to really do wonderful things for this institute and bring the name and fame further so that the country feels proud about it. Thank you.
So now we'll move to today's scientific lecture. So now I will invite our colleague, Dr. Madhuri Subaya, to formally introduce today's first speaker.
You can hold on to your applause because I'm going to really narrate a good story about the great women here. So I'll begin with good morning, everyone, and good morning to our great speakers today. It gives me an immense pleasure to introduce Dr. Priya Abraham, Director of National Institute of Virology in Pune and Professor at the Christian Medical College, Vellore. She is here amongst us today to accelerate us with her talk on our planet, our health, and our future. She graduated with MD and PhD from Christian Medical College, Vellore, in Tamil Nadu. In recognition of her work, WHO had invited her to be part of the team that formulated protocols for detecting and preventing HPV and HIV infections. That's a great honor. Not only she did that, she has served as WHO consultant to Myanmar to formulate hepatitis virus testing guidelines for that country. She was also part of the National Task Force in India to evaluate the molecular epidemiology of human papillomavirus infections in India. She has several national and international awards which adorn her crown. And to name a few, the recent ones, in 2020, she was awarded the Business Excellence and Innovative Best Practices Academy Award by the New Delhi Institute of Management. And last year, she was conferred the prestigious Integrated Health and Wellbeing Council Janani Award for her medical research leadership. So now goes on the interesting story about her. So she was barely 2 months into NIV in November 2019 when the institute came across a sample that eventually was confirmed as COVID-19 positive. Her story goes like this. She was about to catch a flight on January 29th when this was in 2020, when she received a phone call. I quote, "Ma'am, I think we have a positive." So that night she headed back straight to her institute and started assessing whether the new virus has really indeed entered India. After a night full of discussions with her team of colleagues, scientists, she braced herself to inform the authorities that COVID-19 indeed had found its way into India. She had smartly lined up 2 labs for COVID-19 research. One was for the primary lab. The other one was to verify whether the results from the primary lab was indeed correct by a blindfolded manner. And finally, on March 9th, 2020, they were able to isolate the virus, and the rest is history, as all of us know. So I especially got excited when I read about what you said about viruses, because it kind of aligns with me and my excitement about viruses. So she has said, I quote here, "Pediatrics and infectious diseases are my first love, but viruses have grabbed more attention in the recent years." Now that sets a stage for all of us to welcome her with a big round of applause for her and also her team, which really deserve for the work they have done. Welcome.
A very good morning to, hello, am I audible? A very good morning to Dr. Taru Sharma, Dr. Majumdar, group leaders of very prestigious scientific groups, project staff, PhD students, technical staff, representatives from administration, and anybody else I possibly have left out. Today is a very sacred day for your institute. And from what I hear, the founding members really gave of their sweat and blood to create an institute like this. And in that sense, you stand on holy ground. We need to remember that. And I really believe that the strength and the wealth of any institute are its people, not the instruments, not the gadgets, not the nice ambience and space that you have, but the jewels of any institute are its people, and we must not forget that. So I'm here from the National Institute of Virology to try and bridge the interests in infectious diseases affecting humans, affecting animals, and thinking in a hopefully progressive way, where we would finally actually hold hands to bring in the culture of One Health and making our planet safer. So One Health, as all of us know, we are reminding ourselves of what it is an approach that recognizes the health of people is intimately connected to the health of animals and the environment we share. Now, the One Health Zoonotic disease prioritization actually spearheaded by the United States, is a process by which this multi-sectorial approach facilitates this One Health leaning to prioritize zoonotic diseases of greatest concern that need to be, and I emphasize, jointly addressed by human, animal, and environment health sectors in any country, region, or localized area. The emphasis is joint collaboration. You know, it is just like members or the parts of our body. You know, we work, every part of the body works in a complementary manner. And I think the foundation stone of One Health is that all organizations that are dealing with these different sectors must hold hands. And the time is really ripe and really ready for us to work in a very collaborative manner. What is a zoonosis? You all know that. It's naturally transmitted mostly from vertebrate animals to humans. Animals play an incredibly big role. And we know that what infections transcend from an animal source to humans could be bacterial, viral, parasitic, or even fungal, or even an unconventional agent such as prions. Sometimes we know some of these agents can cause a huge public health problem. And in the food and agricultural sector, we know that it can prevent efficient production of food of animal origin, create huge obstacles to international trade, and can actually make a big hit on that country or region's economy. Now, just to recap some of the major zoonoses that we have encountered in the 20th century and earlier, if you go back to the pandemics of the past, perhaps one of the big players was plague itself. It's caused by a bacterium, the intermediate host being a rat. Then there are other exotic agents like Lassa fever, caused by a virus, again possibly transmitted by a rat. Some of you are working with leptospirosis, a bacterium transmitted mostly by rat and other rodents. Monkeypox, now the buzzword, caused by a virus originally found in certain species of mice and rodents, squirrels, yet other rodents to be identified. Crimean Congo hemorrhagic fever, another exotic virus infection transmitted mainly through livestock to humans. And we have Chiasm forest disease originating in Karnataka, but now along the entire Western Ghat region of our country, a viral disease that is mainly occurring in rodents, shrews, occasionally gets into monkeys. So you can see how a teeny weeny microbe like a virus can find its way through human populations, and some of them actually result in diseases that have huge mortality, anywhere above 50%, some going up to 88 to 90%. More of them, we know the first SARS virus, which was detected in November 2002 and then on to 2003. From a bat, definitely, probably the civet cat being an intermediate host. H1N1, actually a combination of pigs and birds. That's how the H1N1 virus came to be. SARS-CoV-2, definitely from a bat. Pangolin probably still considered as an intermediate host. Avian influenza from many avian species, reservoirs being probably in poultry and swine, different types of birds. The MERS coronavirus, probably from a bat, but dromedary camels are definitely an intermediate host. Ebola, not very clear, maybe bats, maybe primates and other vertebrate animals. Rift Valley fever, again from livestock. Zika, probably monkeys. Nipah, definitely bats, and also earlier outbreaks from Malaysia and parts of India seem to suggest it could also be from swine. So we know bats, for instance, are one of the most, I don't know what adjective to give them, but they are the kind of mammals that are capable of transmitting viral infections that we can't even pronounce. And they find their way into different animal species and very successfully transmit infection to the human host. And some of these infections then go on to human transmission. It's believed today that even today's SARS-CoV-2, the culprit of the current pandemic, is originating from bats. So the One Health approach, we remind ourselves of this because we know that in human disease realms, 70% of all human pathogens actually come, 70 or 75% come from animals. And now there is a collaborative effort very distinctly to attain optimal health, both in humans, animals, and to nurture the environment, definitely in some countries. In our country, definitely the Integrated Disease Surveillance Program that caters to human disease reporting, as well as the animal disease reporting program, surveillance programs, need to be intimately enmeshed with each other to pick up the earliest red alert. Intersectorial coordination, which I've already alluded to, really has to become part and parcel of our areas of work, be it humans, animals, and, as Dr. Majumdar said, not to leave out wildlife. The DHR, the Department of Health Research under the Ministry of Health and Family Welfare, through ICMR, is now funding a One Health Center packed in the middle of the country in Nagpur. We have already partnered with MAFSU, which is a Maharashtra Animal and Fisheries State University, and they have been very, very cooperative. We are working together, but this is funded by ICMR, and it's hopefully will be up and ready by the end of next year, latest by early 2024. The pre-project activities are all completed, and now I think they are just going to be publishing the tenders for the beginning of the construction work of all the labs. This will house animal facility BSL2, BSL3, of course, administrative and training blocks, as well as a BSL4. So we'll go on to what are we seeing with the trend in human infectious diseases outbreaks per year. In the paler yellow are infectious diseases originating from animals. The darker brown is those that don't seem to have an animal origin, and you can see clearly those of animal origin is only expanding over the last couple of decades, 3 odd decades or so. So I'll just come now, zoom in to the institute I come from, and to tell you a little about our efforts, particularly in chasing virus outbreaks in the country, some of human origin, some of animal origin, and really time and time again we need to remind ourselves there is no divide really. It should be seamless. So we are one of the 27 institutes of the Indian Council of Medical Research. We are this year 70 years old, and in 1952 we came into existence as the Virus Research Center, jointly set up by the ICMR and the Rockefeller Foundation in USA. In 1967, we became completely Desi. We were taken over by the ICMR headquarters in Delhi, and we got our current name. Our full name is ICMR, National Institute of Virology, and that came to be in 1978. From then on, there have been many milestones where we have become a regional and reference center for arbovirus studies. I suppose all of you know what arboviruses are. And we are also a collaborating center with many Southeast Asian countries, an enterovirus research center, which was located in Mumbai. That center has now become one of our ICMR NIAB field units. We have also become a collaborating center for hemorrhagic fevers, and also diseases of high consequence, be it Nipah, Ebola, Crimean Congo, hemorrhagic fever, and other viral KFD, chiasm forest disease, all that are high consequence pathogens because they are associated with very high mortality. We also became centers, national centers for hepatitis and avian influenza. And yes, there have been milestones along the way, but this is a brief introduction. So what has been our mandate? Our mandate has been to study multiple facets of virus diseases that are of public health importance. So we have gone on, once we have investigated an outbreak, to develop our own virus detection and diagnostic tools, cultivate state-of-the-art research platforms for vaccines and antivirals, enhance the public awareness of a particular disease that is originating in an area. And of course, in no institute can there be no forays into human resource development. We have our own master's program in virology. It's a very tough course, very intensive. And we also have PhD students. And we're now trying to introduce, it should be starting very soon, a post-graduate diploma in virology, medical virology. It's a 2-year course, so that somebody who's had a diploma, who's had a basic graduate degree can come and focus only on viral diseases. We also have a short-term course for clinicians who want to get updated on viral diseases. Like, pediatricians have a lot of childhood diseases that are of viral etiology. So they can have a crash course for a month and take a tour of the entire institute and pick and choose, depending on their specialty, what viral diseases they would like to have a better understanding of. So our mission has always been to serve the nation, and we have investigated in the past over 500 epidemics and outbreaks of suspected viral etiology. We have conducted many serological surveys. We have a huge archival collection, over 250,000 serum samples from human, animal, and bird sources. We have isolated 38 arboviruses, 24 being completely novel to science, and we have more than 500, actually today probably we have 900 strains belonging to various virus families, numerous strains of dengue, influenza, KFD, and today SARS-CoV-2. These are isolates that have occurred over the period of time. And during the pandemic itself, we have accrued over 400 different variants of SARS-CoV-2, many of them being variants of concern or variants of interest. So our medical entomology and zoology department, historically, some of the senior scientists will know, was one of our strongest departments, where we carried out extensive phonistic, taxonomical, bioecological, and disease relationship studies on blood-sucking insects and acarines, birds and mammals. Several new species were identified within these bugs. So be it mosquitoes, be it new types of sandflies, sucking lice, fleas, bugs, ticks, trombiculid mites, and also in other kind of mites, we could find new species while our entomology group was very active. We have a very rich legacy in that area. We have also found new species of rodent and subspecies of birds in our pursuits. We have established colonies of several species of mosquitoes, sandflies, and ticks. We have been able to develop excellent insect diagnostics, mosquito inoculation, immunofluorescence assay, ELISAs that are used to holistically investigate outbreaks that are due to these insects that cause several of the viral diseases. And also many insect species from different parts of India have been screened to determine insecticide resistant status. So, we have spread ourselves a little thin, but I think we have definitely contributed to some of the causative living forms that have contributed to the persistence of viral infection in our country. So one such is the Chiasm forest disease. This was first reported in 1957. It belongs to this virus family, Flavivirus. Many other illustrious members, hepatitis C, many other important members belong to this family of viruses. It was found in the Shimoga forests of Karnataka and mainly evergreen and semi-evergreen forests. And occasionally what they found was there was a spillover from the rodents and the small animals living in these forests to monkeys. And in fact, monkeys would be found dead because this virus that is usually seen in forests, this virus actually infects ticks. These ticks infest the small rodents. These small rodents pass on the infection to monkeys. Monkeys have been found dead in forests. That's why at one time they used to call it monkey fever, not monkeypox, monkey fever. And occasionally man becomes also vulnerable to this infection. So how does that happen? And this really happens when we foray into forested areas with deforestation, establishing dwellings very close to the fringe of the forest. Also, very often the village inhabitants let their cattle and livestock free to go into the forest. So they go graze for the day and they come back. These cattle live very close to the houses of the farmers and agricultural workers. And of course, everywhere close to this sylviatic setting, we have monkeys also coming closer to the dwellings of human beings. People pick up foliage, grass, firewood from the forest. And you can see in these images, as people are dealing with all this vegetation from the forest, you can very easily imagine that these ticks will climb onto all of these twigs and foliage. And when they carry it on their bodies or work with it, they get bitten by the ticks that actually carry this virus. So you can see how virtually every part of the ecosystem contributes for with the way it is set that human beings can pick up this infection. So it's not surprising that this infection, which was first found somewhere there in Shimoga, has now found its way along the western coast, western Ghats of India, and even people in Madhya Pradesh and in West Bengal, we have found some amount of seropositivity among individuals, and that probably calls for a study to look even among the Eastern Ghats, and that's something we're going to embark on because the ecology is very similar in the Western and Eastern Ghats. So you can see something which started in a very restricted area, given the way it can find its way into human beings and the way we have been mercilessly bringing down forested areas for our own gain and our own, profit, so to say. It's very easy for something that was in a niche area to actually spread all the way along the western coast. In fact, in Goa and places such like villagers go into the forest to pick up the cashew fruit. Goa is very famous for its cashew nut industry. So they go to pick up the cashew fruit and the nuts. And when they go into the forest, they get bitten. And typically, the Chiasm forest disease outbreaks are between January to June, when this fruit is in season, and outbreaks of disease happens at that time. Another zoonotic disease is the Crimean Congo hemorrhagic fever, belonging to a different virus family. It is a virus that's associated with over 50% mortality. And for the first time in 2020, my predecessors, Dr. Mishra and Dr. Maurya, picked up seropositivity in the livestock, buffaloes, camels, cattle, goats, sheep, in the state of Gujarat and later on in Rajasthan, where we found that livestock were being infected with this deadly virus. So yes, that points to the livestock from Rajasthan. And then in 2011, a year later, the first human case of Crimean Congo hemorrhagic fever happened. And in fact, the first death was a nurse who attended to the first case. We had a cluster of cases A, B, C, D, and E. And the first person who succumbed was a healthcare professional who was handling a case with Crimean Congo hemorrhagic fever, a new infection that nobody was thinking of. It had found its way from Western, Afghanistan, Pakistan area. It's seen in Eastern Europe, in Western Asia. It's also seen in Africa. It has now found its way into the Western states of our country. We were able to develop the diagnostics, not only serology. We have developed serology, ELISAs for the livestock, and we have developed ELISAs and RT-PCR, both the conventional PCR and the real-time PCR for testing in animals and in humans. And we were able to do a quick diagnosis within 15 hours of the first case. Chandrapura Encephalitis originally was found in Maharashtra, but it's reared its ugly head in 2003 in Andhra Pradesh. It's a disease that mainly affects younger children, 5 months to 14 years. It can cause convulsions, vomiting, lead to unconsciousness and encephalitis very quickly, within a couple of days, and you can see the mortality there. It's 54.5%, and many villages in Andhra Pradesh were struck by this zoonotic illness, and it's mainly transmitted through the sandfly. This is not a mosquito, this is the sandfly. So you can see now both CCHF and Castor forest disease by ticks, this by sandflies. We also investigated a canine distemper outbreak in the Asiatic lions found in the Gir forest in 2018. You must remember that even though it's called canine distemper, we of course attribute this distemper virus mainly in the canine species, but it can affect felines as well. And that's how lions became susceptible in the Gir forest, and we were able to investigate it and find out the etiology. We worked very closely with the conservator of forests, the chief conservator of wildlife in Gujarat, in Gandhinagar, as well as the Sakarbad zoo officials, to be able to actually get samples from the succumbed animals, find out the etiology, and then the officials were able to segregate the surviving animals so that there were no casualties. This has been reported in other parts of the world many years earlier, but this was something completely new for our country. So the conclusions was that we were able to detect canine distemper virus using a real-time PCR. We were able to look at the complete genome of this virus, and from about 11 samples, and we found that these grouped or clustered with the East African strains of the virus. It has been reported from Africa before, and I think thanks to the hand-holding between the conservator of forests as well as the zoo officials as well as NIV scientists, we were able to quickly find out the etiology and prevent more animals from succumbing to this viral disease. Rabies, you all know. I presume you have a very active group here on rabies?
Not exactly.
Okay, I think this is a disease that needs more work on, and it's something that shamefully is there in our country even today. You know, the way India is growing, we should be able to somehow reduce the number of rabies cases and rabies deaths in our country. And you can see either through the mainly through the sylviatic cycle, it crosses over to dogs, domestic dogs, wild dogs, street dogs, and finally finds its way to man. And you know that in almost all cases, I won't say 100% of cases, in most of the cases, it's associated with sure deaths, especially if the individual has not sought anti-rabies immunoglobulin and rabies vaccination immediately. We've also helped with outbreaks of avian influenza right from early 2000s, even up to last year. Whenever there have been outbreaks of suspected avian flu, we have joined the central team to go and investigate. And you know, whenever there are suspicions of avian flu, huge hundreds and thousands of poultry are culled, they are destroyed, thrown into appropriate pits and absolutely snapped out. But we, as earlier, we used to study the avian samples, but now you know this, the center in Bhopal should be looking into avian influenza. So we don't step on their toes, but we do take blood and throat and nasal swabs from the human beings who handle these possibly infected birds, because they can pick up the infection. And so we do their testing, and if they are positive, at least today we have an antiviral in the form of oseltamivir that can be given so that this cannot become fatal in them. But we did have one case, the first case of avian influenza H5N1 in a human being, which we reported last year, the first time in India. It was a child with blood cancer who went to AIIMS for treatment, and he developed fever, cough, coryza, breathing difficulty, pneumonia, went into shock, acute respiratory distress syndrome, and despite mechanical ventilation, they lost him. His sample had flagged as a group A influenza, untypable. So they sent out the samples to Pune, and sure enough, we found it to be H5N1. Whole genome sequencing has also suggested, of course, we looked at the HA gene also, and has pointed to it being an H5N1. Luckily, in India, so far, human-to-human transmission has not occurred. But this is something we should be watching this space, because ever so often we have outbreaks of avian influenza. Another virus, which maybe some of you have heard of, transmitted this time by a mosquito, mainly the Aedes species, Aedes aegypti and Aedes albopictus. This virus is transmitted to the bite of the mosquito, causes a very innocuous disease. You know, nobody gets very sick, but it causes a very silent, very undesirable outcome in a lady who is pregnant, who is carrying a baby and has a developing fetus. Because if this infection occurs in a pregnant woman, the infection is transmitted to the fetus, and what it really causes is, this is the normal sized brain of a young child, and you can see the brain is much shrunken, what we call microcephaly. So when the brain mass is shrunken tremendously, you would imagine that the brain functions also are that much compromised. So this is something no mother would want to have to suffer, because the size of the brain and the head has been so much, reduced because of this viral infection. And it's particularly critical if the mother has this infection in the early stages of her pregnancy, because that's where organogenesis is happening very actively. We have seen that outbreaks of Zika have been happening in the last few years. NIV, National Institute of Virology, was the first to detect the first outbreaks in 2017-18 in Gujarat, Tamil Nadu, Rajasthan, and Madhya Pradesh. Last year, we had for the first time, cases being reported in Kerala, Maharashtra, and Uttar Pradesh, and this year again in Maharashtra. And studies that we have conducted following this recent detection of Zika virus infection in most states, both from South and Northern India, has found that activity is also there in Telangana, in Jharkhand, in Punjab, and in Delhi. So one very good activity of the National Institute of Virology is that through the support of the Department of Health Research, we have built up a huge network of virus research diagnostic laboratories. Some of you may have heard the acronym VRDL laboratories. These are labs that have been trained to detect a variety of infections, high consequence pathogens, as you know. So when SARS-CoV-2 struck, by February first week, we had trained 13 labs in the VRDL. And today, all 139 can do the assay we have taught them. And that has been expanded to more government laboratories, over 1,300 laboratories. 60 to 70% of them use the assay that we taught them. So this is a very good network. Dr. Kotoch, it was the brainchild of Dr. Kotoch in 2013. And it came into existence in 2016 in a more full-fledged manner. And these, the location you can see are strategically located. Some are closely located close to major airports too. So even when we reported the first SARS coronavirus, we trained the VRDLs that were closest to the airports because we knew it's going to be imported into our country. Likewise with monkeypox, we knew it is going to come in from outside. So we trained immediately when we got our first cases, the VRDLs closest to the airports, but we have representation in every state, including Jammu and Kashmir, to kick into action. So 132 labs have been trained for Zika virus surveillance, because of the very undesirable effect it can have on pregnant women. And in the last few years, we have been also battling the outbreak of a very dangerous virus known as Hanipa virus. It belongs to the family Paramyxoviridae. And we know that the early outbreaks first was reported in Malaysia, but the early outbreaks were seen in Siliguri and Nadia. And you can see the kind of mortality. 68%, 100%. And then after a gap, in 2018, we had our first major outbreak in the state of Kerala. 18 individuals were found to be infected, of which we lost 16, leading to a mortality of 88%. In 2019, we had a single case, but because by then, most of Kerala and our 4 VRDLs in Kerala were quickly trained to do point-of-care testing, we were able to detect quickly, provide a supportive management to this young man, and he survived. But in 2021, we were not so lucky. We lost a young adolescent boy to Hanipa virus. And you can see that, you know, what started in Eastern India is now seen in Southern India. And what has been a little scary and which has come to light is that definitely one of the reservoirs are fruit bats. Okay, at least 3 species of fruit bats. And there were many lessons learned and definite trainings given to people after these successive Hanipa virus infection. In fact, that was triggered off after the 2018 outbreak. We gave training in risk communication, contact tracing, not to panic, but to sensibly do contact tracing, bring in effective hospital infection control, give intensive training to healthcare workers who may be, you know, the first point of contact, and nobody knows that's a case of Hanipa. So we need to teach them general practices of biorisk mitigation, and also if they see a suspect case, how to handle that sample carefully, how to package it carefully, and make sure it's transported and shipped to the referral lab, that is NIAB, so that nowhere along that chain anybody gets infected. We also have highlighted the need for hospitals and laboratories to have an adequate stock of personal protective equipment. That means protective gear when you're handling encephalitis of maybe unknown etiology, or a disease of unknown etiology, which could turn out to be something like this. And also, particularly because the state of Kerala was very badly hit, there is a huge amount of awareness of this disease. We have taken every effort to strengthen government laboratories and particularly the BRBL network to be aware of the clinical presentation of this disease and how to carefully handle samples, handle the patients who are suspected of having infection and sending the samples for referral testing. We also prepared several set of guidelines, hospital infection control with reference to a deadly virus like Nipah virus. We also put in clinical management guidelines, how to handle a dead body who succumbed to Nipah infection, the safe burial of dead bodies, sample collection and transportation, the clinical case sheet to tick the different presentations which might point to this disease. And of course, all these guidelines have gone up onto the NCDC, the National Center for Disease Control, which is the main hub of the Ministry of Health and Family Welfare. They work through the IDSP, the Integrated Disease Surveillance Program, to disseminate information that there is an outbreak in some part of our country. We also went on to developing self-sufficiency in, chasing bats whom we suspected were possibly the primary source of this infection. We developed our own bat catching apparatus, that is a Pteropus medius bat that our own staff have carried. You can see that personal protective gear. They've captured it and they take throat swabs, nasal swabs, rectal swabs, and blood samples. Sometimes animals are naturally dead, so we take these samples in a liquid nitrogen container so that we can sample the tissues later. And none of this work, we have already completed bat studies in 10 states of India. None of this is done without getting a priori permission from the conservator of forests. Remember, when we go foray into these lands and do our bat trapping, catching, sampling, the custodians of that forest land must be in the know. And we always get the animal ethics approval from our own institute. It's very important because if things go wrong, then it will be too little too late. So we need to go through the process of getting all the permissions. And we found that many of the bats were seropositive and some of them actually had Nipah virus RNA, suggesting that these are reservoirs of infection. Now we come to monkeypox. We all know, in fact, monkeypox is more of a misnomer because the virus actually comes from small rodents, Gambian dormouse, squirrels, other small rats from the Central African and the Western African forest land. It occasionally gets into non-human primates or monkeys, and it can get into, it got into the African human population very often when they have eaten improperly cooked bushmeat that they pick up for their, livelihood. And then, of course, secondary transmission, we know, is very rampant, and that's how you have a cluster of infection. Occasionally, these African animals, in fact, the Gambian dormouse was once exported to North America, and that those infected the prairie dogs of North America, and people who were handling prairie dogs picked up monkeypox. So you see how one rodent species that crosses the seas to another continent then infects a local rodent species, and or which some of them are reared as pets, and then it crosses the animal-human barrier. These are some of the images of monkeypox from the first 2 cases in India. As of now, we have 9 cases. Again, we have kicked into action by training. I'll come to that. This is again by first, when we noticed monkeypox rearing its head in Western Europe, going to United States of America, going to Australia, going to UAE, we started with our preparedness. We started working on our diagnostics so that if it were to come to India, we should be able to detect it. And that's exactly what we did. And that's how we were able to detect the first few cases. The first 4 were in Kerala. They were men who came back from UAE. The cases from Delhi do not have a history of travel, but they do have a history of, being in these big rave parties with other men. So that today, monkeypox unfortunately has got some stigmatization because it's seen mainly in men. It's seen mainly in men who are sexually active with other men. So, so what we have done in these last few weeks is that, of course, we established testing. We got positive controls from the EVA in Europe. And once we got the positives, we were very sure that we were testing right. We became the central laboratory for testing. We initially trained 15 critical labs close to airports, went to 19. Today we have 32 labs. I think 2 more, 2 out of those 32 need to get the reagents, but we will have 32 labs trained. We have handed over standard operating procedures, reagents to them, including reagents for other viruses that can mimic smallpox. So typically we will have a virus like chickenpox, which is not a pox virus, it's actually a herpes virus, chickenpox resembling monkeypox, herpes simplex virus infection resembling monkeypox, an enterovirus infection that causes hand, foot, and mouth disease resembling monkeypox, particularly in young children. So we have not only equipped them for monkeypox testing, but to do differential diagnosis testing. So many of the so-called monkeypox were actually false alarms. They turned out to be either varicella zoster positive, few herpes, and in children, many of them being hand, foot, and mouth disease. We have given them, we have given reagents, SOPs, controls, and trained the VRDLs as well as 7 Southeast Asian countries. We have succeeded in isolating the virus. It grows very well in Vero CCL81. We have published an expression of interest in from ICMR headquarters for any biotech company or vaccine manufacturing company to come forward so that we can make diagnostic tools as well as vaccine for monkeypox. Apparently, there are 15 bidders now as per yesterday. Yesterday was the last day. So we will now be having a screening of all the bidders. We will be commencing validation of all the commercial kits. Just like SARS-CoV-2, we have a lot of suitors who come and say, I made a very good monkeypox kit. But you cannot take any kit that you get off a shelf. It has to meet the validation criteria. So we have established validation criteria by constituting a national committee. We showed our data. We showed our reference panel, and now the committee has convened that we are ready to do validation for all these commercial kits that are coming into the, who are wanting to get their kits marketed. So coming back to the theme of this talk, our planet, our health, and our future. Health, we all know from a human realm, is a basic human right. And when any country or any region is doing well for health, it is definitely a marker of sustainable development. But increasingly, we humans have embarked on unsustainable practices that have placed pressure on our planet, whether it is resulting in erosion of soil or pollution of water or air, whether it is increasing emissions of greenhouse gases, the deforestation that we do, the land use change that we indulge in constantly, our contribution to climate change, which we do not realize actually impacts on our health directly. We always say climate change will lead to changing ecosystems. But for example, if we look at what is happening in Delhi, we all know almost 1 in 7 people in Delhi have serious respiratory diseases because of the pollution, particularly in some months of the year. So climate change and pollution impacts directly on human health. We also have ever expanding urban areas, which are denuding the rural parts of our country. Introduction of non-native species of animals into different parts of the country. In some parts, fortunately, not so much in our country. This preference for exotic meats and the thriving of wet market trade, which is very common, particularly in the Southeast Asian countries and in China. Inadequately planned development of water and land resources. We build on wetlands to meet food and energy needs, and we know what that has happened. It has caused immense flooding in cities, and then you have outbreaks of mosquito-borne diseases, leptospirosis. So it is a chain reaction. And surely we would realize that nature is slowly revealing to us that there is an interdependence of environment, animal, and human health. And it's also reminding us that we are running thin on our resources. I have this list of other zoonotic diseases is surely not complete, but you can see in all the 4 verticals. And if you were to have a 5th vertical of unconventional agents, we have a whole slew of zoonotic diseases that these are well recognized, and there are many more novel agents which we don't know of. We have detected a number of new novel viruses, but we have not been able to yet see any public health importance. But all of these have at some point in time impacted on public health. The emergence and re-emergence of zoonotic diseases is something that we are seeing particularly in the last couple of decades. And I'm just giving a case study of KFD and CCHF, Castor forest disease and Crimean Congo hemorrhagic fever. Both do not have effective antiviral drugs. KFD was identified way back in 1957. And CCHF was first identified in India in 2010. And both have continued to re-emerge ever since we have detected them in our country. Both are tick-borne, one transmitted by the Hemorrhagic tick, the other transmitted by the Hyloma tick. And this is where the One Health approach is extremely important. Early detection, developing quick diagnostic tools, and responding and quickly to manage the disease or outbreak that ensues. We have to identify all stakeholders when these kind of diseases keep re-emerging. Capacity building and developing cooperation with these stakeholders is absolutely essential so that we can completely understand the epidemiology and the geographic spread, including its spread into domestic animal reservoirs. Because in our rural and rustic settings, the livestock lived in very close opposition, juxtaposition to the hut in which the farmer or the agricultural worker lives. We have to develop a laboratory network across verticals in our country. We need to provide diagnostic reagents and train all stakeholders in biosafety and laboratory diagnosis, expanding that network from there. Some of you must have read about this virus. This is a Hanipa virus. It's in the news as of last couple of days. Shrews are possibly the most likely reservoir. 5% positivity in dogs and 2% in goats. 35 human infections. Infection begins with fever, cough, headache, muscle pain, nausea, vomiting, loss of appetite. Nothing specific, but it causes a reduction in white blood cells, platelets, eventually leading to kidney and liver failure. So far, no mortality. So far, no human-to-human transmission. But we need to be watching this page. And again, the first of its kind being seen in China. And I think the journey will continue. This is our original campus set up in 1952, the historic campus. It's a heritage building. This is our newer campus located in Pashan. in Swiss Road. And located in this campus is the iconic BSL-4 building, where we have done all our kit development and testing for the most dangerous of viruses, even suspected yellow fever and Ebola, which were not eventually yellow fever or Ebola. We did all our preclinical trials for the Covaxin, SARS-CoV-2 vaccine in this iconic structure. And I think we are here to continue to serve the nation. Thank you for your patient listening. Jai Hind.
Sure. So, thank you so much, ma'am, for such elaboration of what all massive work that NIAB does in the background to keep all people of India in peace possible, or how can we, how NIAB contributes to keep Indian public health in a better state. So now the floor is open to question. Madam will be happy to answer questions from the students.
I think there's a question here. She's been raising her hand for some time.
So I'm actually wonderful to enjoy that. So my question is, you beautifully laid out what was done for Hanipa Virus. So it is a non-aerosol transmitter. So it was, we were able to curtail the infection, and I think Kerala government also won an international award for it. But can the same model be adapted for other viral diseases, for example, the monkeypox virus, and prevent the spread? What is your thoughts on it?
So I think, you know, the first two important approaches to curtail the spread of any infection is one, to develop diagnostic tools and to delineate the case definitions. And the second arena is to educate. So already, unfortunately, monkeypox has got some stigmatization. People are associating it with gay activity, sexual activity within the same gender. So that, you know, this, when we go into that space, like it was for HIV, we have to do that very tactfully. You know, we cannot say, you will get it if you have sex with men, men have sex with men. No, we have to do it with sensitivity and care, so that people will not suppress the information. If they suppress information, then our efforts will go, waste. So every disease has its own unique approaches, but it's always multi-pronged. And yes, it is possible. And our first efforts was to strengthen the laboratory testing capacity. So because we train now the Delhi lab, now the Delhi lab is able to report its own positives. The last 2 positives Delhi detected on their own. So they immediately isolate the patient and then do contact tracing. So from the laboratory side, we are here to, first of all, note the clinical presentation and apply the appropriate tests, and also have the armamentarium to detect viruses that can mimic monkeypox. You know, because otherwise a person can be wrongly labeled as monkeypox positive. So definitely the fact that we have done a reasonably good job, WHO approached us to train 7 Southeast Asian countries. So we send them the reagents, the SOPs, the positive control, and train them online. Now everybody's become Dada in training online. So we train them online.
Thank you, ma'am. It was a very informative talk. I have 2 questions. One is, what's the biology of bats and rodents, and what are the traits that make them the reservoirs for so many viruses? And the second question is, are there any studies going on to identify the unknown viruses that are already residing in the bats or any other reservoirs in India?
So bats, there is something about their immunology, and we've not really forayed into immunological studies in bats. They are reservoirs of a zoo of infectious agents. And the funny thing is they do not become sick due to most of those infections. Likewise, most reservoirs, particularly most intermediate hosts, they don't really often become sick. They are, transmitters of infection, and unfortunately it lands up in the dead-end host, which is a human being. With regard to your second question, yes, we have been, I told you we did studies for Nipah virus in 10 states. We also looked for coronaviruses in bats. And we have found that, yes, there are coronaviruses, not SARS-CoV-2, but bats have their own species of coronaviruses. And as you know, even if you look at the horseshoe bat, which is, that's the bat that has coronavirus, which is the most closely related to this SARS-CoV-2. So yes, we have those specimens, and we will constantly be expanding, the list of agents we will be screening them for. They are known to transmit Hendra virus also. That was noticed in Australia and in Singapore, I think. So yes, that's very much on. And it's a big process to, first of all, get permissions from all these forests and also track them. So the latest is we are going to be using drones to help us, of course, getting all the permissions to catch the bats which are very high up in the tree. And once we get these specimens, we will be expanding the list of viruses we'll be screening them for.
What is the trait in bats?
They are often not symptomatic. That's exactly what I'm trying to say. Okay, they're reservoirs, but they're not symptomatic. What made us think of even fruit bats was the first case of Nipah virus was a person who was running a fruit and juice shop. So we were thinking, did he eat some, half-eaten fruit? We could not get, of course, the virus from fruit, but we started, and that's how we got positivity in the bats. The bats often are completely hale and hearty. They don't have any suggestive symptoms that would say, oh, this is a sick bat. Then that would be very easy for us to get them.
Thank you. So I think what Karen says, I hope just to what you do.
Give her the mic.
They are electrocuted a lot.
Yes.
And they are the ones which have been told that they do harbor the viruses. Especially as she says, one of the fruit bats. Some work which was going on back time, I have personally noticed, especially in the late evenings, they come and they prefer the guava trees, So I hope you may agree to that. And one very interesting thing, maybe not much of studies which have been done into the bats, one peculiar paper I came across and read, and that says that especially the pregnant bats, they have the capability to continue keeping this, they would continue this pregnancy for long, and the virus load would get increasing. And that was very kind of new and pretty scary to read about. And there are different kind of bats and their habitats and their work, and they would just dig in near the fruit trees. The big trees, they would just stay inside the crevices of that. And as she correctly says, and everybody knows that she has authority to say, actually, we all only read it. So they just carry and nothing happens to them. And so, as you were saying, maybe, I don't know, would you like to, just with saying that, my point would be, would you really like to advise or suggest a little more focus on this bats, not only food bats, but in general, there are, I think, 36 or 37 species, and they have their own characteristics, their own timings, and I think some are very big ones and so on. So I don't know, what is your opinion about it? Because we don't know that particularly keeping these ones, as one of the slides was showing that, yeah, they are, and we all have witnessed. So what are your views to that?
So actually, as we speak, I've been signing permissions at the institute level for our staff to go and trap more bats. So we don't kill them, we just take specimens from them. But like Dr. Sharma had mentioned, some of them may die due to causes unrelated. Then we take the whole carcass in the liquid nitrogen and come back. In the Northeast, they have shown that bats are a source of rabies. Nimhans in Bangalore has shown that the bats in Northeast, and I don't know which species, I cannot recall, carry rabies. And in Northern America, people, who go hiking and trekking and they spend the night in caves. And if those caves are particular, there are cases of rabies infection being transmitted to individuals who have slept the night in those caves. So yes, it is a sort of a area which is enigmatic. We need to really delve into this. There may be more viral infections, more bat species that will be harboring some dangerous viruses, maybe other bacteria, who knows, even fungal infections. So yes, that's an area that's on our minds, and we are actually expanding our surveillance in other states of India.
You can answer.
So thank you so much, ma'am. It was wonderful. I take your attention towards the wild animals that you spoke about, the Canine Distemper, which moved into these. So I was working with wildlife very closely before I moved here. So we have a lot of such infection happening in neurological part. Like they started talking about parvo virus.
Yes.
It moves very easily from the animals which are closely domestic dogs and all. And now recently, we have this SARS-CoV-2 again. This is also being said that it's moved. So my question is that we were trying to develop diagnostic for these cases, but the positive samples, as you said, control was very difficult to find for them. So we run some tests so that we have a guideline for that. So does NIAB actually provide such kind of samples of wild animals also?
So the virus is not going to be very different no matter from where we source it. So the same RT-PCR can be used, and we can give you controls. In fact, during the pandemic, we got some tiger samples to test for SARS-CoV-2. I think even some gorillas that came from Singapore, because people were talking about reverse zoonosis from humans, animal handlers to animals. If I'm a zoo handler and I've handled many species of animals, that it could go into a new species. So we do that testing ever so often. I think they're not finding anybody else to do it, so they send it to us, because really we are supposed to be now in the human domain, and animal domain should be taken care of by Nishad in Bhopal. But I think that has to be augmented, that we should have a kind of algorithm that certain infections should go there. With avian flu, it's well established. And certainly, if you want to have some positive controls, we can give you positive controls. You can look in different animal species. RT-PCR is the same, and we target three segments of the genome, so that even if there's variability, conserved regions of the genome. So even if there's variability, we will not miss. We should not land up with false negatives. So and in fact, all our variants, even when we came up to Omicron variant, we've been able to pick up because our primers are targeting very conserved regions of the virus genome, and we target 3 different regions. So we are able to pick up.
So the challenge has always been, to isolate these viruses. We don't have that facility, right?
Unless you have a level 3, we actually isolating in level 4, because this is extremely transmissible infection. So we've taken it to up, because we are lucky to have a level 4. So we handle it. Our isolations were all in level 4. And isolation is like you've hit the jackpot. Now with monkeypox, so now we will make our own diagnostics. And whoever's been shortlisted for vaccine manufacture, we will work with them. We've also revived our own old vaccinia strains going back to 1973 and 1996 strains. The vaccinia virus is now growing in our facility. So we could even go back to the original, Edward Jenner technology of using vaccinia virus to protect against monkeypox.
Ma'am, I just wanted to ask your opinion about the same bat or the zoology. I have a pre-perception of mine that when the virus comes from the lower and lower rodents, either it is because of the higher defense, immunity defense, that the virus is just infecting or it's not showing up the pathogenicity, or it is just using the ticks or any rodents as a host just to replicate and to get out, or both it is doing. Basically, it comes to the bat. When they are infected with many bacteria, viruses, and everything, they also develop an immune system against it. So that it have the immune system to fight it, but not to eliminate it, so that the virus, there is no symptoms, but it is replicating and just coming out.
You're very right. One is that the immune system of the bat probably has a way to fend off these infections, and there's an optimum, virus load being maintained, almost like a commensal, in the bat, not causing disease to that reservoir host. But when it jumps to a vulnerable population that has no ability to mount an immediate immune response because they are a naive population, then it creates havoc. So I think I'm sure there's something in literature about the bat immunity that allows them to harbor so many microbes and they don't fall sick.
So to this adding a point, we human beings should also be boosting our immune system as much as we can. Instead of taking the more of antibiotics for the small illness, which can be a helpful in developing our immune system.
Yes, there's been lots about this area during the pandemic, how to stay healthy, how to naturally boost your immunity, starting from a good night's sleep. Something as simple as that, because when you erode into your sleeping hours, your immunity comes down. Eating healthy, exercising. These are all simple ways by which you keep your body tuned and in optimal health. Eating right and healthy. All that is, like now there's a big campaign about increasing the protein in your food. We Indians typically have a high carb diet and high fat diet. So we should have a high protein diet.
So,
yes.
Like for the final thing, we cannot reverse whatever we have done to the nature or something. So we are in my expectation that one thing or the other thing, either the pandemic or the we are about to check. So considering all the bioinformatic tools and everything, then we be looking at a mutation that any virus can come up and we can be ready of it. Like considering the COVID coronavirus, every year we have seen a new serotype. So it is like, can we do it in a bioinformatical level, considering like out of the having a disease of genome, out of that there is 100 or 10 or 50 probability of mutation that can come up. Any opinion?
So actually today, some of the good offshoots of this pandemic is our whole genome sequencing and bioinformatic strength has really developed in our country. We already have INSACOG, which is the Indian Consortium for Whole Genome Sequencing of this virus. NCCS is a member, NIV is a member. We started as 10 government laboratories, key laboratories, and today we are 52 laboratories. And when we got the monkeypox, we did whole genome sequencing. And we found that there were certain SNPs that were seen in certain regions of the virus, which could probably, it's too early we need to do more animal experiments also and see, that is probably contributing to its slightly different clinical presentation this time round. So I think the tools in our country are not lacking. We have enough tools, but we have to be always on surveillance mode. I always tell my colleagues in NIV, we are civilian army. We cannot rest. Any new infection, our antennae should start bobbing and we should be investigating. But when I speak in the context of One Health, that proactiveness, that passion should come from all the sectorial sectors of health in our country, not just human health. It should be animal health, wildlife, and also taking into account what we are doing to the environment that could favor the crisscrossing of infections across species.
So I have a question. I really enjoyed your talk. And what I found fascinating was that you said there's almost a zoo of different types of viruses and other microbes in the bat species that you profiled so far.
Yes. I mean, I won't say in the 3 that we looked at, I won't say there's a plethora, but bats generally, if you were to look into literature, have a zoo. What we have looked at, we have found 3-4 agents, but we have not looked for other agents. The fact that, whether there are bacteria or something, we've not looked for. And that's an answer to the young girl's question, that we need to be, expanding our investigation tools.
So that is what I was going to take forward from her question.
Yes.
Was that, are there any tools, for example, say using machine learning or artificial intelligence based tools to predict from the genomic sequences of these different species that are now being identified and complementing it with the say susceptibility genes of humans to predict
what could be an outbreak in humans? Yeah,
outbreaks in humans in the future.
That's a very important area you brought to light, and people are getting interested in that arena because predictions are so important today. Because human beings are sitting ducks for so many infections we have not experienced. So, using these deep learning tools, if we could make predictions and, look at the epitopes that these viruses express, and then see what could be probably something that humans will not be able to tackle. So it's a very important point. I think elsewhere in the world, they are doing that. We are foraying into this for SARS-CoV-2. We should actually be doing it for other viruses. Yes, the future is in that. Absolutely.
Thank you.
Thank you. I had one key takeaway that I took from this was not just the health departments, the administration and the government also needs to be in tune with the scientific community, because they are the bridge between the scientific community and the government. So on that note, you mentioned that KFP remains endemic in the sea rats, especially among farmers who live with cats. And you also mentioned that you do not have a drug yet. So I was just wondering, the fact that they remain endemic, is it a correlation to the fact that we've not been able to find the medicine? Is it because they continue to evolve the mutating? And am I right in assuming that antiviral drugs also in this case include vaccines? So are there any vaccines against KFP?
So yeah, thank you for asking that question. We actually handed over strain of KFD to the Karnataka Animal Husbandry Department, Veterinary and Animal Husbandry Department, way back in 1998. That vaccine actually was, at that time, formalin inactivation was not considered wrong. Today, most kill vaccines don't use formalin, formaldehyde as an inactivating agent. We have gone on to things like beta-propiolactone. So that is a space where we wrote a letter to ICMR to publish an expression of interest for that as well. And I've been brazen enough, I happened to meet the Bharat Biotech International Limited when we had a joint book release. But, very often the problem is, see, we are not a vaccine manufacturing center. I will say this loudly, and I hope there is some representation from pharma, but unfortunately not here. But many pharma companies look at the bottom line. What is the commercial value of that vaccine or that, ELISA or whatever testing assay that they develop? They always look at the bottom line. Why suddenly monkeypox has come? Because think, abhi to zyada testing ho jayenge, to hame bhi. Coronavirus, people went mad. You know, in a day I'll have 5-6 suitors saying, "Hamara kit, hamara powder, hamara ointment, hamara lotion is antiviral." You know, people were so insane. Just out on a lighter vein, there was one gentleman. I don't have my ID tag, but he had an ID tag. I won't tell you his name. He's an ex-IITian. Sorry. Okay. He came with this sachet of powder, antiviral powder. And he was so strident to me. He told me, "Yadi you take this sachet and show, Corona aage nahi aa jayega. Do rahe." You know, so people look unfortunately only at the bottom line. And KFD is a vaccine that is waiting to be, a newer version of the vaccine has to be made. We are ready to give the strain. We have this live strain. We can inactivate it. But pharma companies also don't come forward because in their perception, it's in only one part of the country. Utna, yes, it does. Antivirals are harder to develop, but we're going to work with Tata Institute of Fundamental Research because they are showing some interest. We will be working with them on what they think. They have developed an antiviral and we have the virus, so we will work together. So that KFD, maybe antiviral will happen. Vaccine ought to happen, but people are not, interested if it doesn't have, in their perception, sufficient commercial value.
Okay. Yeah, that's a very good talk. Right? Just because it is scientific question which emerged, and two of them are thinking in the right direction. So one of the things is that I have a question. First of all, on lighter note, and Madam said, you have to eat well, sleep well, and if you think bats are the right guys, you have to fly and sleep hanging upside down. Probably will be said. And on a serious thing, that when you catch these bats, do you think that in some particular organ there is more concentration of the viral load than compared to any other? Have you ever done it?
We have actually the whole idea of taking the carcasses back was to look at different organ species. We have found virus load, not huge numbers, but viruses in the kidneys, in the lungs and liver. We have found in these three, but not aggregating, like in a skewed manner.
On which organ?
Like it's been, many of them have died probably due to unrelated causes also. So it is, it may be.
They might not have died, as you were saying, that they're carrying, and most of the carriers, as we know, virus will go only inside the cell. So they have to have receptor. Otherwise, it will not go in, because if it doesn't go in, it will not duplicate. But the only thing is the intracellular machinery, which doesn't allow it to replicate that much, as much it would do in human. So there is a restriction in the CRISPR-Cas9 has come like that, for example, or the avian influenza originally has made a chicken where siRNA is made inside the cell.
Counter the virus.
Proteins. And whenever virus tries to replicate a lot, it doesn't allow, because it will not allow it to make a form. So probably those kind of mechanisms
do exist
in the bat cell, which doesn't allow it to proliferate much, but it's kind of a symbiosis,
yes.
kind of thing. So that may be a very interesting thing for people like you who may like to take certain organs which are predominantly allowing this virus to go back. And you can do a lot of in vitro studies to show comparative studies.
Absolutely.
Find a mechanism, but still it won't give you a drug.
Yes.
Because human beings.
But it's good to know the mechanisms, and would be good to address what Madam had stated. Yeah. Dr. Bapat had stated, to make predictions. Right. And what is it in bats which we don't have, which would make us vulnerable, using these advanced tools. So
thank you very much. I forgot that the first director of the institute was Professor Redanna. So we must remember him also, because he's the one who got the land and created everything. So it's my mistake. So I have to catch up with that. Thank you.
One more question. This is the last question here. After that, we will all give the key. Yeah. Okay. Wonderful talk, man. I just wanted to know about your views on that. Recently we have been hearing about the gain of function research, right? So, as a virologist and a researchers, So what is your view on that?
Actually, we have not forayed into that. And most, I think more than the bats and the virus, the group of people, I mean, what I have been fighting are the press. And they're always trying to get the wrong word out of my mouth, to see whether we are doing that kind of work. And they're always wanting to know whether this was an intentionally made virus, genetically manipulated. I think that's where you're taking me. They keep saying, are you sure it was a zoonotic spillover? It was an accidental leak. And we know that WHO has finally been trying to address it. They've had 2 rounds of investigative teams. The pros and cons have been discussed. And right now it's still leaning towards zoonotic spillover. But yes, I think as we foray into more and more advanced tools, get fascinated by the genomics and, the cellular functions of what we understand from the genomics within cells and within microbes, I think there is a big responsibility that rests with labs, including an institute such as this. Where when we play with or foray into genomics, we should do it with responsibility. Likewise, we have the live stuff with us. We need to do it with great responsibility because, it is so attractive because you have grown the virus, you've got the whole genome. It would be so interesting to just tweak it a little. So sorry, we do not have any expertise in that area, and I am not qualified to talk about it. Meaning, I really don't know much about it.
But have you advocated it, or you don't want us to do so?
I personally am not an advocate.
Okay.
Okay. Thank you so much, ma'am. So we could actually see the excitement of talking about viruses and how gracefully and with so much of clarity, madam has given answer to all the questions that we raised.